that's a front row
Good there?
question.
Okay. Let me know when you're secure and I'll...
Maybe we'll ask again later.
Okay. Did that, and I think we got enough reach to reach the sponge now, so this is going to be perfect.
Turn on the lasers again.
Lasers?
Yeah.
Ruin the beautiful imagery.
True.
Kidding.
All right.
Are you happy for me to zoom back on Argus?
Yes, you can zoom on
Cool.
Argus.
Thank you.
There's your laser on sponge.
Yeah, she's big.
Yeah.
It's like, what is that? 10 or 15 lengths of that 10 centimeter, 150 centimeters.
As soon as I get down and around this rock, I'll
One
some more.
You know, Trevor, you got the geologists are really big rock, you know. Just saying, that's all. I'm kidding. We don't need this one, I know it.
We can strap it to the top.
Yeah, totally. Backpack.
Oh my God.
All right,
I mean, this thing's got racks, right?
Am I allowed to kill the lasers for some more
Yeah,
imagery?
Sure, yeah.
Yes. All right, you can come down a lot in your delta tube, please. Okay. I think I can fit between it and the rock.
This guy is called Calophacus subgenus oxydiscus. And it's an undescribed species.
Now this is one of the hardest things for a video engineer to image because it's so bright and the background is so dark. So we'll see how it goes.
Well,
Challenge mode.
It's pretty nice so far right now. You
Yeah,
can see some of the pores in the side of it. I mean, the Argus is just outstanding. Just to see and
Wow, that's a really beautiful picture.
wow, we finally saw one of these whoppers. Wow.
That's a whopper, all right.
It's a Jurassic sponge.
It is.
How old is this sponge?
Oh my goodness, I have no clue, but I don't think it was born yesterday.
Neither was
Wow.
It's unknown, but I mean, this could be potentially hundreds of years old, I suppose. I have no idea. Maybe even older.
Oh,
Look at the wrinkles on it.
Yeah.
Amazing.
Actually, when they get old, they get more and more wrinkly. We've seen the various life stages of this particular species. They start off as kind of little bumps, and then they kind of sort of get older and more wrinkled as it gets.
And it's quite clean too. It doesn't have a lot of that brown kind of you see in some of the other sponges, like in the Faraday ones, where it gets all brown and almost algae covered on the back. It looks like some kind of fur almost.
Yeah, this is all alive. There's no dead parts to it.
No,
it's thriving.
And
flying, Trevor.
this
Again?
top
Nice
of
flying,
the
looks
sponge, would that be pretty solid to be able to hold it? And
So this is not
Yeah,
what's
great
called a
flying,
hard
Trevor.
sponge. This is a soft sponge, even though the stalk can feel pretty stiff and hard. But if you were to sort of squeeze the top part of it, it would be spongy like a kitchen sponge, this particular one.
Really?
Now here's a perfect example. As you see, we were talking about
Such a great
bolosoma
shot.
and how the stalk attached to the convex side of the sponge. Here's Calophacus,
You can
and
push
the
in a little
stalk
bit
is going into the concave
there.
side of the
Right.
sponge.
Oh, the squat
But just
lobster.
a little detail like that can get you far in the game of IDing what it
wow.
is. And you see these
looks-
openings right here. Those go all through the little bends there.
yeah, that's incredible. Nice pan, ROV team.
I wonder if you could put the lasers on now because you could get an idea of the actual diameter of this head,
There you go.
wherever they are.
Top left corner of it.
Oh, jeez.
That's a good scale.
That's
there,
got to
isn't it?
be like a meter
Oh,
across or
geez.
something? That's
Probably, yeah.
ridiculous.
Wow.
Wow.
That's incredible.
Look at the veins on the top right there. Oh, incredible.
Wow.
Can I
Right.
hug it?
I don't think you can hug it. I think you're going to
Wow.
need a ladder to climb it.
It's like
Do you want
this
to push in more
Wow.
to see some of the
umbrella.
structure
So how
on it?
tall would that be?
I don't know. I
That's
don't
so
know.
pretty.
Meter,
3
How long
meters?
is
5
Hercules?
meters?
3 to 5 meters tall.
Yeah.
Yeah, yeah.
Look at the web. Incredible.
Yep.
That is fantastic.
It's otherworldly.
That is unreal.
Well, Chris, I think you just sleep for the rest of the cruise, and we've achieved the apex of spongedom right here.
I doubt if I'm ever going to see a bigger one than that one. That's the king right there.
That is.
See if we can find that little squat lobster on the right side of the stock. See if we can say
Oh,
hi.
there he is.
Yep, there he is.
And that's a little Munidopsis, I think. There, they typically are white, and they kind of have blunt claws.
Well, that guy is obviously the king of this surrounding kingdom.
Wow.
He's got the ultimate condo.
Yeah, no kidding. Penthouse suite.
Uh-huh.
How long did it take that little squat lobster to crawl up and find his home? Wow. Oh my God, there's 100 stairs in this thing.
Wish they'd fix the elevator.
Really?
And what is it eating there?
It'll
Is
just
it
get
coming
it'll just get stuff from the water too. And the sponge is filtering, right? So the water is going to be coming through that hole. And
Wow.
I don't know which direction, but
All right, Aaron, you want to let it leave
I
frame
think
before
the water
zooming out?
comes in that direction, the way the crab was, and then comes out
Keep it
these
zoomed
pores,
in.
but I'm not absolutely sure.
Wow.
But so do these sponges have little cilia
Yeah,
that
alright.
move the
Come on.
water through
Well
the
done,
sponge
Trevor. That was
like,
amazing. Fine. Really
Thanks,
nice.
that was awesome.
sponges do?
Good work
Good
on that.
teamwork.
They all
Everything.
do.
They all do,
It's
yeah.
the cell.
Yeah.
Really.
Yes.
I can back away from the screen. I'm like 2 centimeters from it.
Oh, all right. Where am I? What am I doing? Where are we?
Doesn't matter.
I've forgotten everything.
That was
Yeah,
great.
we've achieved the apex of our
I'm looking
existence.
north. What? I don't understand
So
where I am.
are these
Okay.
sponges common to this area? Are we going to see another one?
Well, we can see some more of this species, but I don't know if we're going to see any bigger than this. This is pretty amazing. But beat that, coral people. Beat that.
Wow.
I don't even know what to do next. I don't know.
I don't even
What
know.
are we doing?
Yeah, I think let's just recover.
Steve has accepted the challenge. He's going to go for those Iridogorgia, I'll bet, on his watch.
All right, I'll just go towards Argus. When in doubt, find Argus. I don't know. There were some cool other alive things over here.
Small things.
Yeah,
Well,
no,
this
everything
is clearly
looks
the
tiny.
contour
now.
to follow, or the slope thing. Not that that means we can do it, but we should do it.
Oh, I'm glad we really saw
Asako
one of the live
Matsumoto
ones.
enjoyed that view too. You know, she's a coral people. I mean,
Uh-huh.
yeah.
You're back to fully white on Argus.
Thank you.
A viewer commented that a water flow study on this sponge would be spectacular.
Yeah, absolutely.
All
Yeah,
right.
they have done
Shall we
this
try
for
and track
smaller
along this
sponges. They
contour
use
then?
that green fluorescent dye,
See
Yeah,
how
I'm
and
looking
they
at
try to
that super
see
cool
how much of that is taken up
Is
and
about
by the
285
sponge
is
absorbed.
completely
Yeah.
looking
makes it
at
to the other
the
side. with
Yeah,
it.
that footage is really
completely
incredible.
perpendicular to the thing.
Yeah.
Yeah.
So
So I want to be more like
yeah, obviously don't go
One-
285.
yeah,
but
like 190, 185, so that
Yeah, I
I'm
think
bringing you
so.
as you slide along.
I think that sounds good. Yeah.
Alright. Bridge nav.
190. Yeah, that sounds good.
Standby. Sorry, 190, correct?
190 is good. Yeah.
30 meters, 190. Thank you.
Try and stay along this kind of ridge drop-off interface thing to see if we get more of this, more lovely treats. To be honest, when I went over the stock, I was a little terrified. I wasn't 100% sure.
I know,
I
right?
was going really slow, so if I tapped it, I could dodge, but I know, it's so scary.
So
I
a
would
lot
love
of
if someone finds the stills from that and actually does the measurements on it. I would love to hear how big it actually was.
Mm-hmm.
Exercise for viewers at home.
So a lot of the species that we are observing here are endemic to these locations, right?
You know, that's the whole problem with the DC, calling something endemic when we really haven't visited very many seamounts around the Pacific. That's hard. Like you go to a site like this and you see something really unusual, you know, and then you think, oh, it's an endemic. We've never seen it before. But maybe you've only visited 50 out of 50,000 seamounts in the Pacific, and you just have been on the wrong seamounts, and it could be elsewhere. So it's really tricky calling anything endemic in the deep sea.
What a weird wall.
Oh,
I know. Maybe the next one could be like 225 or something.
Okay.
Yeah, I think you might be thinking about the shallow waters where we have much better data on the fish and the other species. Even in the mesophotic zone, we have much better ranges for those animals than we do for these.
You can come up on Delta a little bit.
Papahānaumokuākea in general is one of the highest rates of marine endemism for the shallow water species, the mesophotic species.
Excuse me a second. Can you zoom in on the sea star eating the bamboo coral?
Absolutely.
Right there.
You didn't disturb its lunch.
I think this is the first clear case of predation.
Uh-huh.
And if you can actually look underneath it, you might be able to see the inverted stomach of the sea star around the branch.
Try and start with that. That sounds...
Yeah, that sounds tempting.
Sounds terrible.
I don't know how the lighting will be, but we'll see what we can do.
Yeah, it's me. It's pretty
We go
disgusting.
close,
I always
we can open up the iris.
I tease Chris Ma about this all the time. I call his animals evil.
They are...
Okay, zoom in, please.
Yep.
Keep it in frame. I might tilt up a bit more, see if I can get away with going down and looking up.
Yeah, you can clearly see how the stalks were stripped of the coral flesh below the sea star.
Okay, push in more.
What do you think, Chris? Is it
Stomach,
got its
stomach, come on.
extended? I don't necessarily. It's hard to tell. It would be in the center there, right on that branch that it's eating.
See the
Yeah,
flesh.
so all these bare
Yeah.
branches, it's pretty clear he's consumed them all. Yeah, it's hard to see his stomach because his arms are kind of all wavy.
Yeah.
So I think this guy
Check
is
out the
called
top of him anyway.
Evoplosoma, is the species. He's in the family Goniasteridae, and he's a well-known evil sea star.
Mm-hmm.
And that's what he does for a living. He just, what are those big polyps next to it? Look at that thing. Anyway, I got distracted. But anyway, he's cleaned off all of these branches. Yeah, it looks like Evoplosoma.
Okay, come wide, please. So,
And Asako agrees.
did you say evil?
Evil. Yeah, they're eating these beautiful corals. That's evil.
But is that actually the name of it?
No, it's Evoplosoma, not evil. But maybe it should have been named Evoplosoma.
that's what I heard too, evil. That's what I wrote on my notes already. Thank goodness you have your gallery here because I get in the name.
Bridge nav, 30 meters,
So what does
225.
EVO?
Evia, I don't know. But I have my handy Andy Latin roots book right here with me.
Hey.
Let's see what what it says.
Also, how long do you think it would have taken that star to get that much, to eat that much of the coral?
A long time.
So they move really slow.
Yeah.
Okay.
That guy's set for life, I think.
Really?
Yeah, he could just stay on there and probably spend his whole life on it.
No
not
way.
eat it.
Wow, okay.
Evolute and unrolling. That's not right. Eve, good or well? No, because this guy's evil, so it can't be just Eve.
Mm-hmm.
Oh, Ivani, making trouble easily.
Ah, nice. You weren't kidding. I'm going to poke over at this little...
Yeah, this is
rock.
pretty
Yeah,
nice.
Oh, there's one that's, what's this one? Oh, no, that's the wrong word. Okay, now we're getting into some bamboos here. Some more bamboos. Yeah, Asako's mentioning, we see the evil coral beast all the time. Yeah. She didn't say evil. I added that to her message. There's some pretty decent sized bamboo colonies here on the top of this ridge.
Ah, nice.
See a brittle star bailed.
I always wonder how long it took them to get up there and just to throw it all away when we come back.
Never mind.
6 months or a year to climb up that high. Like, oh, bye.
Yeah, look at Timmy. What an idiot. He's just bailed. And they weren't even close to him.
Did you call him Timmy?
Yeah, Timmy. Anthropomorphizing brittle stars.
Yeah.
Now we need a,
Timmy
need
the
a
brittle
far
star.
side for the deep sea.
I want to go back to that wall edge.
Cool.
That's where we found the sponge. Might find more fun.
Like
Kind of right this way. I think.
here-ish. If we can get that far. It might
Where?
be too. It was on this
Oh, yeah.
contour.
Yeah, okay. I think it's like right here. I
Yeah.
want to say there's a cliff, but it's also the ridge of it is like going up,
Yeah.
I think. I hope.
Yes, it is the ridge, but yeah, it's confusing because it would look so steep before,
Yeah.
and then we
And it-
moved just a little bit, and,
And it goes away and
I can't wrap my brain around that.
it's not quite linear.
Oh, there's a salps.
You know, I
There's
can't
another
believe, Trevor,
crinoid.
that you haven't said anything.
Said anything about what?
That red coral up there.
I was looking at this crinoid.
Where's the crinoid? I don't
Is
see
that not what that is? The
Is it
purple
hanging off
thing?
that coral
Or is
there?
that an anemone? I can't tell.
Oh,
You can zoom in
yeah.
on that, please.
Oh, good
Yeah, what is
eyes.
that?
I can't believe you hadn't said anything, Chris.
I didn't see it. I was looking at the coral.
Oh, that one's pretty.
Oh,
that's
wow.
Really
That's a crinoid. Is it a crinoid?
Senior moment. I know what this is. It starts with a Z. The hang on, gotta cheat a little bit. Let's see here. That guy is.
Cool.
Here we go. That guy is a xenometrid, and its name changed a little bit, but now it's known as Sathyrometra, we believe. It's common throughout the Pacific, at least that genus is. They're always perching on corals, and unfortunately I was looking it up and I couldn't show you something about it, but they have what are called prehensile cirri, which are their legs that
nice
they're able to
collection.
wrap
Hmm.
around the branches.
Huh.
You know, and some crinoids don't. Okay, Trevor, what do you think this red thing is?
Oh, it's going to be salp.
Salp, okay. Yeah, good guess.
So is this a coral or a sponge?
What's that?
This
Oh no, it's
The
a feather star. It's an echinoderm. But if you zoom in on this, I think you're going to have your beloved bubblegum
I
coral.
know, I didn't want to call it. I wanted you to take my glory. Okay, zoom in, please.
Sure is.
Look at that.
Wow.
You
All
can't
the
tell me they
polyps
didn't
are
look
out.
very similar to the last ones.
Yeah, I think could be the same species, but this guy is all extended. The other one, if you remember, was all closed up.
Yeah.
That's what I was saying. Yeah, totally.
Oh, okay, sorry.
Yeah. The polyps were all shut down and then it looked more bubblegummy.
Yeah.
I like how they color match those basket stars.
Yeah, that's a good point. The baskets are, you barely even see them.
Yeah.
He's moving.
Yeah.
Little leg
Ooh,
poking
there's
out.
a creepy little tentacle sticking up.
Can you come down in delta, please?
Roger.
Wow, what a beauty.
All right, there we go.
So you think this is like cinnamon or cherry or strawberry flavored? I don't know.
Strawberry, I think.
Strawberry, yeah.
Okay.
It's cool. You can really see the 8 tentacles. What do they call them?
Yeah, it's an octocoral, so because it has 8 pinnate tentacles. And you can almost see the little branches around on each of the
They
tentacles.
extend out quite far.
Yeah.
Yeah. Almost fractal like.
Polyps are huge.
Mm-hmm.
So these the balls on the ends are because you have instead of just having a polyp lined up along the branch, you have a ball at the end, and then you have this, like 5 to 10 polyps all grouped in a ball right at the tip of the branch, and that's what forms the ball there.
Yeah. Does that give it more opportunity to catch prey?
That's a good point, because you're getting more mouths out there right at the end, right closer. That's a very good point. I never thought of that before. That's very good. I think it could be onto something there. It forms a better fish net, if you will.
Yeah, it's pretty cool.
Some more of these stock chrysogorgids. I think Steve had an idea of what some of these were, and I'm not very good with the chrysogorgids. I don't know if this is the one he was thinking might be something called flavescens, chrysogorgia flavescens. I'm going to try to look it up.
What are those like white looking dots all over the rocks? Or is that just
Could they be old
sediment?
bases maybe?
It could be. It's worth a quick look.
Yeah. All right.
Worms?
Yeah, they could be old bases that the actual coral's broken off from.
Oh yeah. Yeah, it looks like.
Well,
Not totally sure.
I'm not totally sure either. I think it's worth a close-up.
Okay, you can try zooming there. Kind of stretched out, but we'll see how it goes.
Oh yeah, looks like bases.
Yeah, I think that's what they are. The broken off. Yeah.
It's like a graveyard then.
Yeah, a graveyard. They all broke off very close to the base there for whatever reason.
Mm.
So are you able to tell from this if it's a sponge or coral base? What's a good way to tell the difference?
I'm pretty sure these are coral bases because that looks like it's calcified, whereas the sponge are going to be silicified, if that makes any sense.
Silicified.
I guess I don't know if that's a word. I guess it is.
I don't know that word.
But yeah, I think those are coral.
And what's the distance between the lasers again?
10 centimeters. I almost think it's worth taking a look at this sort of frilly white one that's kind of big. It could be one of those metallic, well, maybe it's what, it might be one of those Ramula, whatever they are, plurigorges. Yeah, never mind. I think that's what it is. We've seen a lot of those already.
Probably get another ship move in.
Sure. If you want.
Sure.
Bridge nav, another 30 meters, 225.
You want to look at it while we're here, Chris, or keep going?
Sure. I mean, I can see. Well, let's go ahead. Let's zoom in
Okay, go
anyway,
ahead.
just to make sure. Yep, it's just a really big colony of one of these things.
Nice.
And again, see all the polyps are lined up in a row on the branch?
Mhm.
All the
Oh,
same.
yeah.
Just
Oh,
like
that's the
military
militaris.
formation.
Yeah.
Yeah.
Okay, come wide, please. What's that kind of yellow spot? Just about top of the screen, middle.
Yeah, what is that?
It's going to be the end of my
What
leash.
about
but-
this thing right here?
Yeah.
Could it be a beloved sponge?
It might be.
You say it might be a type of sponge?
Maybe.
I'll have to wait for a little more. Oh, just kidding. I'm behind.
You're going to get
ours.
yanked right off.
Well, let me pull in then. I was facing the wrong way. I'm sorry. Turned around. Zoom in, please.
Oh, mineral deposit.
You called that out. We didn't.
Okay, I'm going to go. I'm definitely facing completely the wrong way. So, yeah. going to move.
Well, let's see. I've got
That's why
a,
you questioned if I wanted a ship move.
I've got a rock mollusk. You have
Is
a
that a
sediment
question as
creature.
much as
Uh-huh.
a question?
You've got an orange rock creature.
Sure.
Just
Sure
me being
thing.
like, what?
Ooh, this
Where
is
am I?
looking pretty neat.
Going for a ride, buddy.
Look at the density, how it's starting to pick up.
Uh-huh,
Along
it
the
definitely is.
Yeah, this wall
Right for the
feature
end of
is-
your watch. Sounds about right.
Yeah.
But we had a pretty.
We had a
good
pretty
one.
decent watch. I got no
Definitely.
complaints.
Yeah, we
You
got
got
the
your
giant
giant sponge.
I got,
Yes.
well, even quite a lot of corals. It was really beautiful.
And the little crab with the backpack.
That's right, yeah. So it looks like the wall is kind of this way. I mean,
What are we at now?
see, like kind of this strike here.
Yeah. It is. Yeah, that makes sense. But it is still going up and over too. So
Totally,
it's,
yeah.
like visually breaks my brain right now. Tired.
Yeah, I'm right at the end of my leash.
Um,
I'd have to go down the
what's that?
wall.
Yeah, could we look at this thing over to the left that's just going off screen? Right there.
Yep.
And it is
Good for
another
you.
type of sponge.
That's pretty cool.
Right there.
It might be bouncy. I'm at the end of my leash, but we'll just do our best.
Dead.
I just wanted to confirm, this is something that's called Tretopleura. It's
Half zoom, please.
Tretopleura.
very
Tretopleura,
cool.
and we used to
Yeah,
call
good
these the
there.
big leaf sponges.
Oh, wow. Yeah.
Big leaf sponge.
Yeah.
Cool.
So that's the first one we've seen on this dive too. So again, we just documented another species here.
Uh-huh. That's really
Yeah,
neat.
terrific.
All right, flyby imaging.
That was enough though. That was
Yeah.
just to make sure that's what it was.
Oh, look, overhang here. I'm underneath an overhang.
Ooh, look at
Classic.
that. That's looking
I'm really into
spectacular.
this wall here. Can't go that way. I gotta go away from the wall. I think we need a ship move towards the wall.
Towards the mall? All right,
Yeah.
direct west.
We got some more
Yeah.
of those white
Bridge nav.
paragorgids.
Mhm.
Those kind of thicker branch.
Can we
Yeah.
get a
Maybe 20 meters at least.
20 meter step, 270? Thank you.
Wow, look at that. That's beautiful.
You see how they're right under the lip of the wall. So if you go further down the wall, you're not going to see very many of these things. They kind of hang out right on the edge.
Oh, look at that Gorgidade coming
Oh, wow.
straight
look at
out.
that guy.
Oh, pretty.
I saw some neat shadows. I'm like, I might be underneath something, and yet I still
Whoa.
am.
Wow.
Oh no, I just might
I
have,
think you
That's
might have
above you.
got a little piece of it.
just
Oh,
a little floaty. That is really out there. Wow.
Yeah.
Huge.
Ship's still moving south a little bit.
Roger. Can you zoom in, please?
Nice. Perfect.
Look at that.
oh.
That is stunning.
You never get tired of these things.
No.
It's got to be one of the most beautiful creatures in the animal kingdom, if you ask me.
Mm-hmm.
Just something else.
I really want to look down it again. Oh. Those are some big corals now.
Oh.
Yeah.
Wow.
Is there a reason why they like this type of, I guess, right off the cliff
It's the
environment?
the currents bring food,
Okay.
and they want to be, so the faster the current, the more
More
frequent food, more abundant food.
Yeah, that makes sense. They're pretty big too.
Yeah, they're real big, and so they're doing well. They're thriving if they're gonna, because they're old and they're obviously being successful. They're getting what they need.
Yeah. Oh, thank you.
Oh yeah, there are some big ones up ahead. Look at that.
Unfortunately, the way I'm looking is not ahead right now.
Oh,
Kind of doing some side action.
Just want to let it settle out.
What was that?
Do you just want to let it settle out as they switch in?
Yeah, that's the smart thing.
Okay.
Yeah, I could call.
See all the particles in the water column.
Mm-hmm.
That's all the stuff the coral would eat. Is that right?
yeah.
Buffet.
Certainly some of it. Who knows what that is? Marine debris, or not marine debris? No, they're not eating garbage.
Snow.
So yeah.
What's the term for it? The acronym? particulate matter. No, POM, P-O-M, particulate organic matter. That's what it's called.
So we have a question from the audience.
Uh-huh.
So just why do we not see any coral rubble here compared to other places with previous corals?
So for one thing, it's pretty steep. So whatever dies, it might just fall straight on down and it's out of view.
Yeah, that makes sense.
That would be one explanation for it. And then we're a little bit off the wall, so it might be a little bit hard to see at this point. But it's a good question. We're not seeing a lot of dead corals like we have seen elsewhere as well. And maybe it's just this is a more suitable environment, and they're hanging in there. There hasn't been that much.
And like we have seen a lot of dead sponges, right?
Yeah, up sort of on the flats, a few of them, but not tons. We've seen a few of those stalks.
So one of our viewers did the measurement of the giant sponge.
Did he?
Well,
what did he get here?
Sponge end was approximately 1 meter wide and 0.8 meters tall. I think it was just the
just the ball. That's what he's talking about.
just the ball, not the
yeah. Way to go, whoever you are. That's way to go.
Excellent.
Great.
3 meter wide top
That's
to that
incredible.
thing. That's unreal.
And somebody else said nature fireworks.
Yeah, that Borgia. Yep. Well, we're in the process of a shift change. I'm afraid we're going to have to yield our seats. But no, that's okay. we just couldn't see any. We're going to leave the rock collecting to you now, so you can pick what you want. And we're good. We got some sponges and some corals and things. So, all right, everybody. So I think you're all going to hear us twice on this particular dive because it's a 24-hour dive. So every group's going to be back a couple times, and we'll all see you again. Our group, we're signing off, and we'll see you real early tomorrow morning.
Aloha.
Good evening, everybody.
Check, check, check.
Check, check. I can hear you. I can hear you as well. Great. Okay. Catching up on the chat here. All right, as we're getting settled, remember we are going to take a moment to ground ourselves in silence. Are we ready? Front row?
Yes, sorry, I was off SPL. I hear you now.
Ready.
Thanks so much. So at this time, I'm going to invite everyone to go off microphone. And for those listening at home, we're going to be going into radio silence to conduct a private ceremonial protocol that acknowledges and honors the biocultural resources that are present and that we're interacting with and impacting. We will be back on SPL shortly.
Mahalo.
Yes. All right, Jake. Can I give me some leash here?
All right.
Front row, or Rennie, I guess.
Yes.
Can you tell me how we're doing with ship moves and
Okay, so
Herc and Argus?
we are currently stationary. They were moving along. We're actually about halfway through the dive track.
Okay.
So if it stays a 24-hour dive and we don't want to ascend any further in depth, then we have some time to explore.
Okay.
It seems like the less steep slope has been less interesting for them.
Yeah.
These steeper slopes here is not uniform. There's a lot of these walls and crevices, and moving along has been definitely a challenge, but rewarding with all the corals they've been seeing.
Okay. Yeah, I think especially considering we're making good progress on the dive track, we can just kind of take it slow and check out these really vertical inclines and see what kind of animals we find.
Okay. So for now, do we want to explore where we are? Do we want to move along a little bit?
I wasn't watching the video too much right before this, so I'm not sure how much time they've already spent in this area. Does anybody in the back row have an idea?
I think they were just scanning around.
Okay.
So we can do either. A lot of the fauna we're seeing here has been what we've been seeing for the past hour or so.
Yeah.
So yeah.
Okay, so maybe we should try to maybe move up a little bit. Okay.
Up as in a long slope or upslope?
Upslope.
Okay, we can do that a little bit. You just want to bump up? It sounds like they don't want to, or it sounded like they were finding less interesting stuff up top. So
okay.
I don't want to call too many moves in and we end up in a place where we don't like.
Yeah. Let's see. Maybe let's move a long slope for a little bit.
Okay.
And then if they're not seeing much difference, then we'll slowly start going upslope.
Sounds good. All right. I'll kind of go along the same track they were going, maybe a little more along. How about 225? What's that strike? What's your heading?
My heading's about 300.
300. So if I'm heading 300, then 225 is roughly along that, right? We'll go with that. Sure.
Roger.
All right. I'll just go 50 meters at first, and we'll feel it out. We're a bit shallower than we are last night. So.
Yeah.
Bridge, nav. Good evening, Paul. We'd like to step
what's interesting
50 meters.
about, I was looking at the dive plan that shows the bathymetry for the overall area. This is basically a tongue that comes out of a platform. And it's quite a flat area. And I was wondering, on top of here, there may be a lot of rocky, crusty stuff, but it also is a potential area where we could find pockets of sediment.
Yeah.
So if you want to peek up and do that, I don't know if there's sediment, how much sediment's up there, but it would be worth taking a peek.
Yeah, I think we'll just kind of go along here for a little bit and then maybe rise up and see what we find.
Yep. Jess, I'm going to do a DVL reset if that's all right.
Sure thing.
And it appears that it's really been dropping out, so. Must be pretty deep below us, and that 1200 isn't helping us.
Sorry?
The 1200 kilohertz isn't helping us at this terrain.
All right. And Steve, I'm going to take cues from you on if there are particular animals we're seeing that we should spend more or less time zooming in on.
Yeah, absolutely. We have been seeing a lot of similar stuff. I think we experienced a transition at around 23, where are we now? Around 2350 meters.
Okay.
Yeah, we had something totally different down below, and then we've transitioned to this bamboo coral dominated band. So it's yeah, I'd be interested to see how long this lasts for. You know, as we go up, we're going to be turning over species. So right now we're in this layer where we've had a decent chance to look at everything, but you never know. Occasionally you'll get rare species that pop up even in a community you've spent quite a bit of time in. So we'll take it slow.
Yeah.
And I'll give you a heads up on what I see.
All right. Well, there's definitely a lot of things. The whole screen is full of at least, what, 20 different animals at the moment?
Yeah. It seems like the wall is quite steep.
Yes.
Some kind of
and
verticality to work with here.
A lot of these corals too, they're not fast growers. So this stuff, even though it looks very cracked, and you know, looks like there's maybe gravel and boulder and cobble-sized rocks, it's actually, you know, the biology is telling us that it's really cohesive here.
Yeah, I was thinking that in terms of the rocks too. They don't really look like tailless. They look like part of the original formation. What kind of sampling remains to be done? What are the remaining priorities for us? That's a good question. So far, rocks for the project to look at microbe-mineral interactions on rocks have not yet been collected. And so that'll be a priority for this shift, since that's part of my project. And we were discussing before the shift began to actually maybe try to get 2 for 1, where we actually try to pick up rocks that have animals attached so that I can have that variable as part of the study. And that allows us to collect more biological samples under kind of controlled biobox conditions. So that's one thing we'll be looking for when we find animals of priority for understanding the diversity in this ecosystem. We'll try to, that are attached to rocks and they're not too big to be fitting in a biobox. We'll try that. I think there's still a couple animals on our most wanted list of species we're trying to understand who they are. So if we see some of those, we will try to take subsamples. We still need to take some Niskin samples for the eDNA study. A couple have been taken, but we still have some more to go. But we don't need to be overzealous with this shift. We've got 12 more hours or more with diving on this feature. So we can spend a good bit of time really looking around and seeing who's down here, what the community of the seamount looks like. Sounds great.
One thing apparently they talked about last watch is if we're starting to get towards the end of it and we don't want to go shallow, we could venture down a bit and then track along just perpendicular upslope. So kind of just go down and track up the wall.
Okay.
It would probably, you know, take some time as we go down slowly, you know.
Right.
Hopefully minimal loss of visuals, but
Jeff, you want to do a partial zoom here?
that might not be on our watch, but in the future.
So several different species in the frame of view here. Any of these that we need a zoom in on, Steve?
No, but if we're just sitting here, we can do some zooming as long as the pilots have enough time.
Sure, yeah.
You know, get to some beauty shots, some close-ups.
Yeah,
great.
Jeff.
So feel free to fill the air, so to speak, if we're hanging around.
Yeah, it can't hurt to have more visual identification of what all of these are.
Got some precious corals here, as well as some golden corals, Chrysogorgia corals with associates. There's a squat lobster in the right hand, Chrysogorgia, possibly a Gastroptychus, or some other peristylid crab.
Did you say a crab?
Come wide there, please, Jeff.
Oh yeah, in that, so there's a crab inside that lives inside coral colonies.
Oh, okay.
And they're collectively known as squat lobsters, but there's several different families.
Oh, I see.
There's the Chirostylidae, and then there's the Galatheidae. The Galatheids, you don't usually see them inside the corals as much as the Chirostylids.
Argus has moved about 15 meters of the 50.
Roger.
Looks like we're slightly pulling off the wall, so I might have to descend to still keep it in sight. Try to make the next move back towards it.
Yeah, so we just got confirmation from our lead scientist that we're going to be down here for a while, and
do a partial zoom there, Jeff, please.
Lots of time to explore. Our co-lead scientist. He's the lead. He's got to write all the reports.
With great power comes great responsibility.
Yes. Beautiful rocks in the background, as well as really great diversity of corals and coral-attached communities.
One of the corals we've seen quite a bit of on this dive and tried to sample
All right, full wide there, please, Jeff.
has been these mushroom corals, these red golf ball shaped sized corals. They're quite common at these depths, but unlike a lot of these gorgonians, these branching colonies, they are more, their skeleton is more embedded in the tissue, and they form these small golf ball shaped, we call them mushrooms. That's part of their common name, mushroom corals. But yeah, they can be quite numerous, and they're also one of the few known brooders of larvae. This is why you typically find them in growing in clumps or clusters. You have one large one, and then it'll be surrounded by, maybe 20 or 30 smaller ones. the larvae don't travel as far for a lot of these brood corals. But
Yep,
there were
I'm
studies
getting
off the
tugged a
west
little bit.
coast done on reproduction of these, and they found that they do
Come
brood.
down a little bit more.
That's fascinating. One of the questions earlier, I'm not sure if it was asked at an earlier watch, but was how these coral are triggered to spawn underwater at deep depths. But it sounds like not all of them are spawners.
Oh, we probably just don't know very much.
Yeah, both. The answer is yes to both. We don't know a lot, and it's hard to observe that. But what cues animals to induce spawning and go through the process of gametogenesis and create egg and sperm is different in different parts of the world, and it's different in the deep sea. In the deep sea, in the higher latitudes, for example, it's thought that seasonal dynamics, like spring bloom, for example, when you have a very large bloom of phytoplankton in the springtime in the North Atlantic, it creates a massive food pulse that sends this signal down to animals in the deep ocean to, hey, it's time to start producing egg and sperm. There's lots of nice gooey food. And it's an energetic process to produce those cells. In the shallow water, obviously, coral reefs, it's lunar processes. The corals might sense tidal dynamics or phases of the moon and cue spawning to synchronize with those types of periods.
I'm going to call in a move there.
In areas where you don't have light or don't have
240
any sort of
upslope
visual cue, you have to rely on these non-visual cues like food pulses. So that's the going hypothesis. And I mentioned that it varies across the planet. At the higher latitudes, you typically have higher or more strong seasonal signals, whereas in the equator, or as you go closer towards the equator, there's less well-defined seasons, and so you typically have relatively constant low production throughout the year, and it's also influenced by oceanography to some degree. And so it's not really well known if, for example, as in higher latitudes, corals might spawn once per year in the deep ocean. They may actually spawn less frequently. So they may create their egg and sperm over a longer period of time and maybe spawn every 2, 3, 4 years or more.
You can see how the chances of observing that become very, very low.
And we know this, we've observed this in their tissues, in their skeletons, right? Like tree rings, right? Tree rings are seasonal processes, over the course of a year, depending on if it's wet, dry, whatever the environment, trees will grow more or less. Same thing is true for corals in the deep sea. If it's a particularly good year for food where they can eat a lot and, the environment is favorable, they may grow faster, and this is observable if the corals lay down
there's
annular.
Maybe a sponge, if we could zoom in on that.
Sure thing.
If the corals lay down annular rings, or however it grows. But what we find is that those signs of seasonality and periodicity and growth over years is much less clear as you move deeper into the ocean and as you move towards the equatorial latitudes.
All right, Jeff, go ahead.
It's a cool sponge.
Steve, do you have any thoughts on what that might be?
Yeah, we've got a really nice glass sponge there, or at least a couple of them, maybe. I'm not familiar with this family myself, but we can certainly look it up. It's I don't think it was one of the high priority collections, but
okay.
Yeah, if that's what we're waiting for, I think we can just grab some images and move on. But I'll get back to you and see if we have any better information about identification.
Great, thank you.
Sure thing. Full wide, please.
But it's definitely a glass sponge. You can see the silica spicules embedded.
Yeah. It looked like there was a little sea cucumber off to the right, and we've got another coral coming into the frame on the left. Jess, as we continue to lateral left, maybe if we could start ascending a little bit. Gone down about 20 or 30 meters so far.
Yeah, we're putting in another ship move to get us a bit closer to the coral band as Ernie referred to it. So yeah,
okay.
It just takes a little bit of time.
Sounds good.
Can you hold off on that ship move just a second? Chris wrote in, he actually might want that sponge.
Oh, okay.
Just want to double check before we move out of the way.
Oh, the sponge we were just looking at?
Yeah, the one that's just over to our right.
Yeah, right under that rocky boulder.
Yeah. What would be the best way to collect that sponge? With a slurp or with the coral cutters?
So our slurp is currently potentially clogged.
Oh, okay.
We can try exercising that, but the best one is just going to be to grab it.
All right.
So the way we might want to sample this, we want to get some of the inside of the sponge as well as the outside of the sponge. Sometimes sponges have different morphologies of spicules on the inside of the osculum versus the outside of the sponge. So I think...
Steve, are we definitely stopping to sample?
Yeah, we're going to sample this. Okay. So I think maybe grabbing and...
Would this be a good candidate for picking it up if it's attached to a rock? Or would it be better to...
That might be harder.
Yeah, okay.
Yeah. I think what we can do is just, we have the coral cutters on there, so we can get in there and kind of just chop it in half.
Okay.
And take out a piece.
All right. Thanks, Ernie.
Hopefully that does it. I don't think that you'll ever get pulled off, but we'll find out.
And I'll leave it up to the pilot which one they want to go for, but top or bottom, I think those are two totally different sponges, but they have just multiple oscula there. So what we're looking for is maybe grabbing half of one of those individuals.
Sure thing.
Oh, sorry.
And can we get some shots with the lasers off, please?
Sure.
Before we start cutting.
Jeff, you want to push in there, please?
Might get some with and without.
Yeah, great shot.
Yeah, beautiful.
Actually, I can frame it up a bit better here for you.
Tilt down, or yeah.
Yeah.
Great. All right. Good captures.
Okay. Great.
Yeah. So what I'm thinking, I'm going to draw on the telestrator where I think we